Shaevitz from Princeton is here. You made it. He's going to tell us about swarming and phase transitions in Myxococcus.
This is a very sensitive device. I'll try not to wiggle too much. Okay, sorry, I just got here and I'm in a very different time zone, so I'm going to try to be coherent. My name's Josh, I'm from Princeton. I want to talk to you about the motion of my favorite bacterium, which generated this beautiful plot. And I can tell you about that later if you want. I'll start by thanking the people who worked on me with this. There's folks in my group, and here, Ron Lewis here in the audience somewhere. So if you're interested in this stuff, you can talk to him afterwards. And we have collaborators both on the computer vision side. In Missouri, we work with Christina and Roy Welch, who's here, who's a biologist, and some folks in Marseille. Okay, so this is Myxococcus xanthus. This is my favorite bacterium. Can you still hear me? Okay, good. For those of you who study E. coli, I'm sorry, I'm about to show you Myxo eating it. And Myxo glides on surfaces, and I've been working on it for about 15 years, various aspects of its biophysics, and I learned recently that in fact I was working on active matter. I hadn't known that, but now I know it, and so I guess I get to be an active matter scientist, and we worked for a long time on how it moves, but it turns out that when it gets together with lots of its friends, it does lots of interesting things, and the thing I like about this system very much is it's really evolved to do this sort of collective dynamics. Like, it's its whole goal in life. So it's true that you can take many bacteria, throw them in a little test tube or a little dish and watch them, and if you make them really concentrated, they'll do all sorts of cool things. However, if you look at where that bacterium normally lives, they don't live that way. What I'm about to show you is really what evolution has tuned this thing to do, and we're trying to understand how that works. Okay, so this is a spot, this is an artificial thing, right? This is a spotted colony of Myxo. This is maybe a centimeter on a side, and this is a colony of E. coli, and this is on a dish that has very little liquid, so there's not a lot of liquid on top. It's sort of wedded into the gel, and so these cells are immobile. The E. coli are basically sitting there, and these cells are going to go kill them. And so what Myxo eats are other bacteria. They excrete small molecules that kill the other bacteria, and then they absorb all the good things. And you'll see this front will come over here. It will glide over these E. coli. It'll form this amazing rippling wave-like pattern that I will not talk about today, but is actually very interesting. And then here, these cells have essentially decided life is bad, and they're starving, and they're dying. And so they make what are called fruiting bodies, which are large, intricate mounds. Over here, these ones are going to eat and kill all the E. coli, and then they will also decide that they're starving because they killed all the E. coli, and they're going to form very much these same sort of static things. Within these fruiting bodies, the cells sporulate. It's a very protective state that they can enter into, and they can stay there for a long time. So here are some of these fruiting bodies until you give them more food, and then they will wake up, basically, and crawl out of this thing and go off and do their awesome, exciting, hairy stuff. And this is sort of a low-resolution image. You know, we're talking about many millions, if not tens of millions of cells here. And if you zoom up very closely on different species of myxobacters, these are what the fruiting bodies look like. They're really quite beautiful. So you go from essentially a liquid state into a solid state that looks like this. Now, I'm going to warn you, the one that I'm going to tell you all about today, and the one that we study, is the one that makes the ugliest looking fruiting bodies on the screen. They're just really kind of little mounds. But there are other myxobacters that really make all these beautiful things. And in fact, you can take images of this. I will tell you, so this whole movie I just played you probably takes 48 hours. So these are slow stuff. But we can watch these fruiting bodies being formed, the more intricate ones. And so you can see, this is all from gliding of cells on surfaces and on each other. Okay, and so these are sort of, you might think of as viscoelastic, self-propelled, solid-y things. And they go, and then it's going to explode and make this really cool-looking flower sort of thing. This one. Do I get a cursor? Oh, okay. Yeah, we missed. We got to play this. It's too cool. I cannot play for you. So here, if we scan through this one, we can go shrink. Make this cool little flower. These ones make these cool little flowers too. All right, cool. So this is all from moving of cells. And if you zoom it down, you know, this is what the individual cells are doing. So somehow, by changing the way they move, they're able to go from these liquid states to this wave-like rippling state to this thing that forms a giant flower. And then in the flower, they stop moving. And so it kind of solidifies because they form little round protective spheres around themselves when they stop moving. So there's a few aspects to the biophysics of Myxo that we need to understand to think about the collective motility. One is that they go backwards and forwards. Okay, so they're essentially bidirectional. As far as we can tell, they don't have a head and a tail. They can dynamically change which side of the cell is the head and which side is the tail. And they have a clock inside that they can tune. Okay, and if you see these genes that are labeled FRZ, I know you guys probably hate genes. Too bad. Okay, this is like an actual living thing. It has genes, so we have to learn about it. Okay. There are these FRZ things, and these are homologs or similar proteins to ones you probably have heard about, which are the chemotaxis genes in E. coli. Okay, so E. coli has a chemotaxis circuit that reads in signals and changes the forward and backward revolution or rotation of its flagellum, which makes this run and tumble thing possible. And there's a methylation-based amplification and adaptation circuit in E. coli chemotaxis. So if you raise the concentration of things, it raises the set point of this sort of switch. And so that allows you to adapt to constant changes, so you're only looking at gradients in concentration. That's how E. coli works. And what Myxo has done is taken an amplifier and overdriven it and turned it into an oscillator. Pretty cool, right? And if you're interested in this, there's a nice paper here about how this probably works. Okay, so it goes backwards and forwards, and it can tune this clock. It can tune the amount that you're driving the oscillator. Okay, so it can go faster than slow. So, they're quite flexible cells. They're about 5 to 10 times more flexible than E. coli. And they're stuck to the surface, and so when they run into each other, they naturally want to align. Okay, if they're both crawling along, they will then align with each other and form these sorts of streams. This is not because of adhesion, we don't think. This is really just because they're flexible and they're trying to go forwards, and they are stuck to the surface. They don't like to go on top of each other until they really have to, okay, by buckling out of the surface. And then they have a third interaction, which is that they leave a bunch of gook on the surface, which we only partially understand what it's made of, but they follow the gook of their neighbors. Okay, so if I go streaming through here and Christina comes along my trail, she will then follow me into the pit of hell or wherever it is that I'm going. And so this is another interaction we have to think about. Okay. So let me tell you a bit about the kinds of experiments we do. So we can take movies like this. These are very high resolution movies. This is probably 5,000 cells, and we can use computer vision technology to track them all. Okay, so this is not so trivial, but not so hard. And that we can do at rather high resolution at kind of low densities, right? So they can be pretty packed, but they can't be in many layers and things. And these fruiting bodies that we're going to study and the ripples are many layer phenomena. And so to connect this kind of dynamics to what the populations are doing, we then take experiments where we put in just a few labeled cells that are glowing fluorescently, and we have lots and lots of unlabeled cells that you don't see if you image in fluorescence, and so now we can track their dynamics. So this is a giant fruiting body, right? This is, let's say, 50,000 cells, and where we have a few fluorescent ones there, and now we can track the dynamics. So what are the things we've learned? So the cells can turn off these reversals. They can do it themselves. We can do it with genetics. And what happens is if you don't have reversals, you form flocks. These are coherently moving groups that have all the properties of flocks you've read about in the literature. They have giant number fluctuations. They have all the sort of statistical properties you would get from a V-check or a more fancy model. And so you get motile flocks. And if you turn on reversals so that you reverse about once per body length, as you're moving, so you become sort of diffusive in this really weird one-dimensional way, what you form are streams. Okay, and these streams are essentially stable in space, right? So if you watch this movie, there's lots of things, it's sort of metastable in space, but then everyone's, now they're all gone from over there. There was lots of cells there, now there's no cells there. And this stream here lasts for a long time, right? It's there. Just hangs out there. Okay, if you make a histogram of where the cells are in space, this becomes very obvious. This stream is extremely long-lived. There's basically always cells there during the experiment, and when you have the flocks, they're sort of just going everywhere. Density gets averaged out. Okay. These streams are also immune to giant number fluctuations because they're essentially one-dimensional flocks, which is kind of cool. So if you watch one of these streams build up and you look at the fluctuations of the width, what you find is that they go like the square root of the number of cells there, and they don't have an anomalous power larger than that, like you would get in all dimensions larger than 1, including for two-dimensional flocks. And so when we turn off reversals and we have these big flocks, the group number does exhibit these number fluctuations. And if you look at the motion of cells within the one-dimensional stream, what you find is that it's ballistic for very short times, where it's moving before it turns around, and then it becomes subdiffusive, so that they're really stuck in space, which is kind of nice if maybe you're trying to explore the environment and sense what is over here, or if you're trying to aggregate, which is what's going to happen when they form these fruiting bodies. And it turns out reversals are not all that you need to get these streams. You also need slime following. So I won't take you through all the simulations we did. But basically, this is the one that really will give you nice streams. It's reversing cells that also follow their neighbors with some short-lived slime that they leave onto the surface.
Is there any difference in the amount of slime secreted by reversing cells?
We have no idea. Probing slime is not easy. I wish it was. I would say we have. Okay. So what happens when these cells try to form fruiting bodies? Well, it turns out in very short times, okay, they raise their reversal frequency a lot, which is going to cause this stream formation. They don't change their velocity much. And then at a time of about 4 hours after they've been starving, a couple hours, maybe 3-4 hours, the speed goes way up and the reversal frequency goes down. Okay, and so I can give you a little bit of a sense of the very beginnings of this. The reversal frequency is low, and in fact these things look a bit like flocks. They're not, they're sort of moving all around space. They turn the reversal frequency up after an hour or so, and now you have this sort of long-lived streaming phenomenon. But you don't really get fruiting body formation really starting until about here. And that's the point where the velocity goes way up and the reversal frequency goes way down. Okay, so I want to show you another movie of fruiting body formation now in 2 dimensions. This is just kind of a nice one here. So these things are just starting to aggregate, and there they go. And they all form these nice, wonderful big fruiting bodies with all sorts of beautiful, cool dynamics. If you lower the density, though, and you starve them, what you find is they don't really aggregate. You might see some little coherently moving little groups. These are tiny flocks. Move around. There's a giant collection of tiny flocks that never aggregate, even though they're starving. If you raise the density some on the surface, you do get fruiting bodies forming, and they're forming randomly everywhere. They're nucleating and then growing. And in fact, we have lots of classical phenomena of coarsening here. We have Oswald ripening, so if you watch the small ones, they will evaporate, and their cells will go into the bigger ones. And so eventually, these will all coarsen into a series of very large aggregates, at which point the cells stop moving because they sporulate, so they turn off their motion apparatus. And so I would expect that actually the infinite time limit, if they kept moving, they really would all sort of coagulate into one fruiting body. Now, if you raise the density even more, right, you're going to get classical spinodal decomposition or spontaneous decomposition everywhere. Okay, so a change in the dynamics of what you see. And again, these coarsen, you can see evaporation and everything until the point where the cells stop moving. Okay. You can do lots of sort of meta-analysis of these things. You can look at the time scales over which the length scales coarsen, both for nucleation and growth and spinodal dynamics. You can look at what densities you get a transition from nucleation and growth, which is a low value on this kind of a scale, and spinodal dynamics, and it looks like there's a critical density at which you get this change between the 2 different kinds of dynamics. But we were really scratching our heads, because I'm not an active matter physicist, of what do I make of all this stuff? And then Christina told us the answer, so I will tell you what Christina told me. How about that? And she told me that she and others have a whole theory for how this works. And the idea is you need to think of these particles, right, as the cells as your particles of your material, and you need to think about what drives the fluctuations. And now she's going to be mad at me for this exponent, and I apologize. It's such as life. Okay. And the idea here is that for a self-propelled particle, it's the rotational diffusion that generates noise in the trajectories. Okay, and then you can have them interact with each other, and that gives you sort of a physical system in which instead of temperature giving you random fluctuations, now you have essentially kind of non-random fluctuations because the particles break symmetry themselves. And the thing that plays the role of what you might call temperature, if it was a thermodynamic system, or the thing that generates fluctuations more accurately, is the inverse of the classical rotation Péclet number, which is the ratio of diffusive motion, and I'll take you through these terms in a second, and advective transport. So A here is the length of the cells, dr is the normal rotational diffusion coefficient, and we've done some work with Christina and Lisa Manning to show that if you have reversing cells, you have this term here, which is the reversal frequency. So if you can change the reversal frequency, you can change this Péclet number, and then V is the velocity of the crawling. You would usually call them swimmers, but they don't swim, so they're crawlers. Let's put it that way. And so we know actually that Myxo can control this, and can control this. Okay, so Myxo has the ability to control these numbers, and in fact, as we saw during fruiting body formation, the velocity goes up and the reversal frequency goes down. So if you calculate this inverse Péclet number, it starts at some moderate value and it goes down by about an order of magnitude. Okay, and our question was, does this drive a phase transition, you might say? And so how can we test this? Well, you can do simulations, but since I'm not a simulator for a living, I'll do experiments. And we have various ways of perturbing the Myxo ourselves. So we can make them essentially stop doing what they want to do, and we can either control the reversal frequency, these are experiments we're doing now, and I don't have the data quite to tell you it, or we can control the velocity, and that's the data I will show you, with a small molecule drug. And so we can basically tune the velocity to any value that we wish from their normal one, and we can make it lower. Can't really make them faster. And by doing that, if you make a phase plane between the density of the cells on the surface and this inverse Péclet number, we've so far been able to put some points here. I think there's some new data here that Ron has that I don't have yet. And you can ask, what are the different dynamic phases that these do when they undergo phase transitions? And so the gray points here don't phase separate when you put them at those densities and those Péclet numbers. The blue points here undergo nucleation and growth, and the red points undergo spinel decomposition. And importantly, for a single density, I can get different dynamic behavior, okay, by changing the velocity of the cell. Okay, if you make them go faster, you'll make them move down on this axis. And then if you look at what the wild type cells do when they're starving, they start out here when they're happy gases trying to go hunt things. When they decide that they're starving, they drop their Péclet number down here. Okay, they cross these boundaries and they coarsen and undergo these phase transitions. Okay, so that's all I really have to say. We're doing a little more work here. It's a cool thing. These dotted lines I made up by staring at a paper and then I drew them. I don't think we can theoretically predict them yet. That is our next goal, working with Christine and others. But I think it's a really neat system. And Myxo really did evolve to do this. This is their whole goal in life, is to do this kind of thing. And so I think it's very exciting and fun. And it's nice to see that we have real physics now to describe this. So that's cool. All right. That's going on if you want to ask me questions. Excellent. I think it's lunchtime, so you're all hungry.
Actually, I just want to say one thing. Don't leave, we do have some announcements, so let's ask questions, but then don't leave, please. Questions? Yes,
so you mentioned that the slime was important. Yeah, so you mentioned that the slime was important in your simulations to get the nematic streams. So should I think of the slime as but it's also not a static thing. So the slime forms, but can also die off in some sense. So how much of the say the agar gel would be covered by slime in the experiments?
These are excellent questions. So I would say I agree with the very first clause of your statement based on our simulations.
Okay.
We can't turn off slime experimentally. Okay. The second statement about the lifetime of the slime, we have no idea what it is in the real system. So we can play with it in the simulation. It can't be infinite, otherwise it covers too fast. And then how much slime is on the substrate in the experiment? That's my answer.
Is there, are the slimes all, on average, are they all disordered in directionality? Or?
I mean, presumably because the tracks go
Yeah.
around, but like, I don't know. It's not clear whether there's direction in the slime. I mean, it could be, but we don't know that.
You said that these bugs evolved to be collected. Is there any physiological reason why do they want to do that?
So they have to hunt together. A single bacterium cannot generate enough local concentration of antibiotic to kill its prey. So in fact, they really do need to live collectively, and that's why they both hunt collectively and sporulate collectively, because if they don't wake up with their friends, they're toast, right? So it's, you know, it really is their whole goal in life.
So in your first movie, you show rippling waves only in the region where they equalize. Because they need nutrients, or why only that? I know you said you were going to talk about it.
So we are very interested in rippling. Presumably, so what we found here, right, is that you raise velocity, low reversal frequency, and you drive aggregation. Presumably they tuned some other knobs to make rippling, and they've tuned it on the E. coli so that they don't go anywhere. That's the prevailing idea. So when you do rippling waves, they're kind of mixing things. Yes, because that's where they're trying to go kill things.
So it's not that they need nutrients for cooperation, right? Nothing to do with that.
No, I think it's the opposite. They form rippling waves to get more nutrients.
Okay. Like that.
Hi, Patrick Vanderhill from RPI. My question was, are they doing this on agar? And what's the role of the underlying surface?
So they're doing it on agar because that's the nice thing you keep in the lab. If you go out into almost any pile of dirt on the planet, you will find the fruiting bug. Okay. In fact, the way that you cultivate Myxo in the wild is you take dirt and you stick it on an agar plate that you can't swim on, and the thing that crawls out that's not like a mold is Myxo. Okay. But you find them on dung piles, on rotting tree branches, and piles of dirt all over the place. So presumably the substrate is not, the mechanics of the substrate aren't super important.
So have you done it on other things in the lab, or?
You can get fruiting bud formation, sure, on almost anything. What you can't, we can't image much. We've thought a bit about doing it, but I think basically what happens is in the end they're mostly gliding on each other. Right. So the substrate isn't that important.
Okay. Question
Hi, Michelle Driscoll, NYU. I was wondering, are these 3D in their environment? Like, are you looking at, can you hear me? Are these 3D in their environment? Are you looking at kind of the slice of what happens?
So it depends which experiment. The big grayscale-y things, we're sort of compressing the whole third dimension, but the fruiting body is basically isotropic. To first order, it's
In
isotropic.
the dirt, are they making 3D paths? And this is just like what would happen if you...
In the dirt, it's hard to say. You break open the dirt and you see a little ball, a little orange, and they smell really good. They smell like the forest floor. So as Roy will tell you, if you really love that smell, you're a good Myxo researcher. And the students in my lab who don't like that smell, they work on a different organism.
Hi, Matthias Merkel from Syracuse University. So you've plotted the velocity of the cells over time, right, during sporulation. I was just wondering whether this includes the cells within the spore or not.
So that is the cells mostly on the flat part.
Okay.
In the spore, they go vertically, which we are now trying to make 3D measurements to get real 3D tracking. If you look well, no, let's call it fruiting body. The spore is a single cell. If you look in a fruiting body, just by eye, they all appear to be moving about as fast as they are everywhere else.
Okay.
So it doesn't seem like they're really jammed in terms of stopping the motion. So I think to compare this to the normal two-dimensional models of active matter where things kind of get jabbed up at the aggregates, the idea here is you're de-wetting from 2D to 3D. In the 2D sense, that slows you down, right? Because now you do this and you can't go back up. But that we are hoping to confirm soon.
Okay, cool, gotcha, thanks.
Any more questions? Okay, let's thank Josh again.